All right, kudos to you that's made it in person today. I know it's cold. Time change is a killer. So good job for being here. Before we start today, I had to show you my bracket. Well, it's hard to read. It's like a picture of it in my office. I made this one up with my heart. So there's a lot of Big Ten teams in there. Of course, Michigan's winning. So if you have never filled out a bracket, so many people say to me, well, I don't like basketball. I didn't either until I started doing this. And my daughter is just bananas about doing this. She's a calendar savant. And I think there's something about the way the bracket looks that just, she loves filling this out. And she doesn't even watch games, but she loves keeping track of who wins and who loses. And we don't even play for skin. We just do bragging rights at home. So it's just fun to do. So it takes a few minutes to fill out, you know, whatever, bet dinner or something with your roommate. It's just kind of fun. So what else is there to do, you know, that's fun these days? So you should do that. I think the games start Thursday, or is it Wednesday? Thursday, I think. Anyway, so a week ago, you took an exam. The mean was about 74%. Before I curved, it was almost 70, not quite 70. So I did curve it up a little bit. I did the curve a little different this time. I multiplied the number you missed times 0.9 instead of 1. And then I added a percent onto that. So actually, the more you missed, the more the curve helped you. We still had kind of a lot of D's, but some of those would have been F's without a curve. The person that got 100% had gotten a B on the previous exam. So there is always room to improve. Still got a couple of big exams left. So, you know, come see me if you want to talk about how you're doing. We can brainstorm maybe ways that you can do better. There were 4 questions. There were only 4 questions that less than 50% of you got right. All right, so those 4 questions, this one, which of the following is not involved in the initiation of transcription? So the beauty of taking an in-person exam, right, is that you can go through and kind of cross out the ones that you know aren't correct. Which are not involved. So hopefully Pribnell box, promoter region, -35 region, hopefully you could cross those out. I think a number of you forgot what consensus sequences are. So if you remember this slide, I know it's one that wasn't in the book, but we talked about how the RNA polymerase binds. This is sequences of the promoter region of different operons, how the RNA polymerase binds, especially to the -10 and the -35 region. And the closer that those sequence of base pairs lines up to some consensus sequence, we think the more commonly the RNA polymerase binds or binds more tightly. So the correct answer here was the origin. The origin is where DNA replication begins. All right. Let's see. This one you did have to read really carefully. And, you know, I know some questions ask which are true, some questions ask which are not true. So it's helpful to go through and, okay, yeah, this one's true, this one's true. Okay, so which of the following is false about prokaryotic translation? Okay, so translation can begin before transcription is finished. That's true, because this is all happening in the cytoplasm. Charged TNAs enter the ribosome at the A site. Okay, that's going to be true. The very first one enters the P site. All the rest of them come into the A site. Some proteins require modification after translation. That's true. Formylmethionine is always the first amino acid translated. That's true. D here is the one that's false. Initiation begins with the recognition of the Pribnell box. Okay, that's transcription and not translation. So Again, that one required a little bit more careful reading. We talked about two different experiments. So the Messelson-Stahl experiment, that's one that we said was called the most beautiful experiment in biology, that dealt with how DNA is replicated semi-conservatively. And the other experiment involved Griffin's experiments that he did with those two different strains of strep pneumoniae to prove the existence of transformation. So I think there was a few of you at least that got those two experiments mixed up. That was sort of when I asked a question about both those experiments, and it was sort of like I didn't ask you how they did it. It was just kind of the main purpose of the experiment. So those are sort of, again, don't study the details too much that you forget the main idea sometimes. And then which one of the following antimicrobial applications asked primarily to disrupt cell walls and membranes? Correct answer here is this detergent, quaternary ammonia. So detergents and soaps acting primarily on membranes. And of course, we talked about how that makes them leaky, and that causes all sorts of problems. So, all right, so those were the ones that were most missed. I've only had a couple people come in and look over their exam. So try to do that. I think that in many cases is helpful. Sometimes we can do that by Zoom if you like. So, all right, don't be afraid. Come find a time. If my office hours don't work for you, we can find a time that works. All right, so we had started talking about antibiotics not last week, but the week before. So we mostly kind of introduced chapter 10. We were thinking about how you use antibiotics, like what kind of things you have to think about, like are there side effects? Selective toxicity is the most important thing. We want that antibiotic to target the bacteria and not to harm us. We talked about therapeutic index, administration of the antibiotic, different ways. And then we began, there's about, I think, 14 different antibiotics that I want to talk a little bit more specifically about. So we started doing that, started with penicillin, and that's where we left off. Methicillin, so we talked a little bit more in depth about, I mentioned that we're going to talk about a lot of cell wall inhibitors, but the beta-lactams, remember, they're a big group of cell wall inhibitors that all work the same way. They target those transpeptidases that are necessary to rebuild the amino acid cross bridges in that peptidoglycan structure. All right, so methicillin, this is another beta-lactam. This is what's considered kind of a second generation penicillin. All right, so the original product of that penicillium mold wasn't able to, it's not absorbed very well, and it's labile to stomach acid. So for both of those regions, the original penicillin had to be injected. We came out with, actually, when you grow that mold up on a little different medium, it gives a slightly different product that was able to be given orally then a little bit later. But we came out with methicillin in the late 50s, and originally due to kind of this is considered a side chain. Remember, here's a beta-lactam ring down here. The side chain here, don't memorize the structures, but the side chain is a little bulkier, so we thought that would make it so the antibiotic resistance wasn't gained quite as quickly. That would kind of protect the functional portion there of the drug. So it originally resisted the action of some common beta-lactamases, and I'm gonna show you, talk more about the action of that enzyme in just a minute. There's some side effects. About a third of people experienced kidney inflammation. So this is a little bit lower therapeutic index, meaning, remember, the lower the therapeutic index, the less safe it is. So that's the difference between the dose that works and the dose sorry, the dose that's toxic and the dose that works. I think I said that right. Might have got that mixed up, sorry. So anyway, we had to kind of adjust the dosage a little bit better. So this one, because of those side effects and because it's acid labile, labile means changeable. Let me make sure that I wrote that down somewhere. So basically the antibiotic is changed by stomach acid, so it loses its activity when you take it orally. So this is a drug that had to be injected either by syringe or by IV. And this is a drug that is not even produced anymore. So we don't use it, not even produced anymore. Why am I bothering to tell you about it? Well, there's a strain of Staph aureus that's known as MRSA. Okay, that stands for methicillin resistant Staph aureus. And that you do hear about today, a little bit less so than what we did maybe 10 years ago. But this is a strain, it's named methicillin resistant Staph aureus because it showed up in the early 60s. And at that time, methicillin was still used, and that was a big problem. Oh, here we've got a strain that's resistant to methicillin. We thought methicillin was going to last a long time without organisms growing resistant to it. So the name has stuck. That organism is not only resistant to methicillin, but all of the beta-lactam antibiotics and many other antibiotics that are commonly used. So that sometimes that's not obvious. All right. So those early penicillins were mostly gram-positive, working against gram-positive organisms. In 1971, we came out with a more broader spectrum penicillin, and that's amoxicillin. So again, most of these drugs are, some of them are the natural product of either the mold penicillin or they're semi-synthetic. So we've kind of monkeyed around with a side chain. I believe amoxicillin is a semi-synthetic. Okay. This is one of the more common antibiotics still in use, more of your kind of everyday kind of infections in terms of uses like ear infections, strep throat. Okay, so this is the one that they add the bubble gum flavoring to. So it's probably the most common antibiotic that kids take. You know, I kind of, you know, when my dad, so probably your grandparents' generation, when they had antibiotics, you know, the doctor would inject it. My dad talks about, you know, getting an injection from Doc Martin in his rear end. When I took antibiotics as a young child, I can remember taking them in liquid form, and they tasted like the worst thing you could imagine tasting. Like we'd eat crackers afterwards to try to get the taste out of my mouth. And now you all grew up with bubblegum flavored antibiotics. I mean, you had it pretty good. Hopefully antibiotics will still be around for your children. Hopefully they'll still be effective, but we'll get to that more a little bit later. It had the nice thing that it was acid stable, right? So given orally, it absorbs well from the small intestine. So it gets to the tissues where it needs to be to do its work. You didn't have to, I think this is typically taken like 3 times a day, which still seems like kind of a lot, but less frequent maybe than before that. We do worry about resistance to this. So most of the time, actually today, amoxicillin is marketed in a form called augmentin. So basically that means we add something else to it, this clavulanic acid. This is a beta-lactamase inhibitor. It's supposed to sort of shield the antibiotic so that the organism can't come in here and then beta-lactamase doesn't work. I'm going to talk about that again in just a second. All right, so if you augment something, you add to it to make it better. All right, so augmentin is amoxicillin that has this clavulanic acid added to it to try to extend the usefulness of the antibiotic, make it, again, bacteria less likely to become resistant to it. All right, so I've used this word beta-lactamase a couple times already. So sometimes we refer to that enzyme as penicillinase. It acts on penicillin. So in some ways that name makes more sense. I usually refer to them as beta-lactamases. Remember, these are enzymes that bacteria produce. All right, so sometimes it's more complicated than it originally seems. When we're talking about resistance, remember, we've got our host cells, we've got bacteria cells, and we've got the drug. So it's three different things. So when bacteria, how do they become resistant? We're going to look at some mechanisms of that, some other ones, but one way that they become resistant is the bacteria acquire the ability to make this enzyme. And what that does, it cuts that beta-lactam ring right here. And now that that ring has opened up, it doesn't fit into the active site of transpeptidase anymore. And the antibiotic doesn't work anymore. Okay, so that's one specific way that bacteria can become resistant to a beta-lactam. And we'll pause here for a minute because one of the things you have to think about when prescribing drug is, are there side effects or are there allergies to people taking them? In best number that I could come up with, about 10% of us are allergic to penicillin. And it's not, you touch the penicillin disc in lab and that's not going to hurt you. It's when we ingest penicillin and the metabolized product of penicillin, when it's metabolized in our liver, that's what we're allergic to. And most of the time, it's not a huge problem. Patients get a little bit of a rash, hives. If you've ever had hives, like it's pretty, it drives you crazy. If you had hives like this person, you would be like beside yourself. If any of you know Dr. Baxter or have him in lab, he had a reaction to penicillin when he was a teenager and had hives for 9 months. So hopefully Benadryl did him some good, because that would be pretty bad. All right, so for me, I'm supposed to be allergic to penicillin. I had a little rash when I was like 6, and forever after now, they won't give me penicillin because there is a potential that the next time I have it, I could have a worse reaction. And it might not be shock, but there are a small percentage of us that actually make a certain kind of antibody, an IgE antibody to that metabolized penicillin product. And then that antibody can cause just like system-wide inflammation, or can kind of start that process. So, which can actually lead to shock. So shock is when you have so much inflammation that your blood vessels get leaky enough that your blood pressure goes way down. Okay, and then if that persists for long enough, all your organs can shut down, and that can lead to death. So there are about 300 deaths per year in the US due to an allergic reaction to penicillin. All right, so once you're labeled as being allergic to penicillin, no doctor will ever give that to you, just because apparently the textbook says, oh, you sometimes outgrow it. When I asked my allergist about that, he's like, oh no, you have that for life. And I said, well, will you test me to see if I'm really allergic to it? And he said, no way. He said, we won't do that unless, we have to have that information to save your life. So you know, now I take the $100 antibiotic instead of the free one, but that's not the worst thing in the world, I guess. So, you know, again, it might not be a big deal, but it could be life-threatening. So it's something that we should be aware of. All right, a few other, another beta-lactam that is in pretty common use are a huge group of drugs called the cephalosporins. And these drugs, mostly, there's a ton of them. There's actually 5 different generations. So the oldest ones, first generation, the most recent ones we've made are the 5th generations. Most of them begin with this prefix ceph, like cephala this or cephala that. So that's how you know that it's a cephalosporin. These are produced by a mold called Acremonium cephalosporium. So just a few examples of cephalosporins: cephalexin. The trade name, so this is a confusing thing, you know, you're going into pharmacy school, you know this, but you have what the drug is, cephalexin, and then the trade name, that's the name that the company that produces it slaps on there, is Keflex. Okay, so they're the same thing, it's just, again, the brand name. But you may have heard of Keflex because that's in pretty common use. Again, these are beta-lactams, so they're working the same way that like penicillin, methicillin are. They bind transpeptidases. The toxicity here, it really depends on generation, the target organism, the spectrum of it depends on the generation. The early cephalosporins were pretty narrow spectrum and only targeted gram-positives. We've changed the side groups, and there's a couple different side groups here. We've monkeyed with those enough to make them more broad spectrum, but the later generations are also have a little higher toxicity. So we have to dose them out very carefully. So some of them have to be injected, the ones that have the lower therapeutic index, and other ones we can take by pill form. The higher generations have the advantage of crossing the blood-brain barrier. So we have a few infections that can do that, like bacterial meningitis. So we need a drug that can follow the organism there. So there's not a whole lot of them that can do that, but again, the later generation cephalosporins, and actually, usually if you're allergic to one beta-lactam antibiotic, like penicillin, you're allergic to them all. But the higher generation cephalosporins apparently are different enough that most people, there's only a very small number of people that have a penicillin allergy that can't take those later generation cephalosporins. So again, I don't know when they take that chance and when they don't, but and also the later generation here, the 5th generation, is also pretty effective against treating MRSA. So we still do have some tools there. All right, so still talking about cell wall inhibitors, but now we have a few drugs that are cell wall inhibitors, even though they're not beta-lactams. So vancomycin, you know, if you look at the structure here, it's a huge, bulky structure. It actually is a physical obstruction, still kind of targeting those amino acid cross bridges in cell wall construction, but in a little different way. It just kind of is more of a bulky block there. It is a narrow spectrum, only treating gram-positives. It's probably, again, the first choice in treating MRSA. In general, though, this is a drug, vancomycin, that it's not the first one used. Like you wouldn't give vancomycin for somebody with strep throat. Usually they'll pull out the vancomycin if maybe 1 or 2 other drugs haven't worked. So it's typically seen as more of a last resort kind of antibiotic. When we first started producing this, I'm getting them mixed up whether it was 60s or 70s. We had some problems with toxicity issues. We have learned how to do the organic synthesis a little bit better now, and those toxicity issues aren't as much of a concern now. It isn't absorbed very well from the intestine, so it's always given by IV, except if you're treating C. diff. So we're going to talk more about Clostridium difficile in about a month, but that's a infection in the colon, causes really nasty diarrhea. If you're trying to treat that, you have to take vancomycin orally so it gets to the tissues where it can do its job. Okay, otherwise, anything else you're treating, you're going to want to inject it. So we now have found that there are some resistance growing even to vancomycin. So this gets to be kind of an alphabet soup, but VRE stands for vancomycin resistant enterococci. If you're in lab, you might recognize Enterococcus as being an organism that we kind of group with the Streptococcus. It's really closely related to the Streptococcus. So the Enterococci aren't typically going to harm people that are, not immunocompromised, but this is a hospital-acquired infection that's considered a medical emergency, because if vancomycin is sort of our last resorts, and then you've got something that's resistant even to that, then you've got problems. So bacitracin is, again, interfering with cell wall synthesis. So all of these beta-lactam, anything that targets the cell wall, it's a growing cell wall. If the cell wall's not growing, then the drug's not going to work. So populations that are growing in that log phase are especially susceptible. Bacitracin, so those NAM and NAG sugar subunits are produced inside of the cell, and they have to be transported through the cell membrane to get out to the cell wall. The bacitracin prevents that channel from transporting those subunits. So it keeps the cell wall from growing. Well, or basically there's holes where the new NAM and NAG should be, and right, that disrupts the integrity of the cell wall. It's another narrow spectrum, only targeting the gram-positive. This one is toxic to the kidneys if it's ingested. So for that reason, we only use this one topically. Okay, so skin infections. And it's one of the antibiotics in triple antibiotic cream, or triple antibiotic ointment, whatever. Okay, so which is probably, you get a cut on your skin, you put a little triple antibiotic cream on there, and that's kind of the recommended thing right now. So, all right. One more cell wall inhibitor. And this one's fun to say, isoniazid. Very simple structure here, very narrow spectrum. It prevents the synthesis of mycolic acid. So do you remember what kind of cells have mycolic acid in their cell wall? Okay. Those are the Mycobacterium. Also known as acid-fast organisms. Right, so again, if you're in lab, you'll get, most of us are doing that stain in like the last week or next last week of lab. Okay, this is important because this is the organism that causes tuberculosis, Mycobacterium tuberculosis. All right, so this drug prevents the synthesis of mycolic acid, which is a big component of that acid-fast cell wall of Mycobacterium. So, something like an E. coli, isoniazid, not going to have any effect on the E. coli because it doesn't have mycolic acid in its cell wall. All right, so stepping back, pausing for just a minute. I have found that, at least to me, it has been helpful to make a concept map to think about all these different drugs. So I don't know, if you like brackets, maybe you would like concept maps. This might be a little better option than writing everything out on a flashcard for this unit. I kind of picture this in my head. So you're going to want to focus on the mechanism of action. Okay, so I have those written in here. So the cell wall inhibitors, here's our beta-lactams up here that we're talking about. The book talks about a whole bunch more really briefly. These are the ones that you'll be responsible for. The vancomycin, bacitracin, isoniazid, those are also cell wall inhibitors, just work in a little different way. It's reasonable to know that isoniazid is the mycolic acid inhibitor, but vancomycin, bacitracin, just know that they inhibit cell walls. Okay, and then the different mechanisms here we're going to be talking about next, the protein synthesis inhibitors. So you can fill in the drugs. We're halfway there already, you know, as we go, come back to this. So you're going to want to especially focus on the mechanism of action and then any advantages or disadvantages to using that drug. So I'm mentioning other things like, oh, this is how we administer it. And, you know, sometimes I throw in some other things just so you get used to those routes of administration or understanding the therapeutic index, or, you know, whether it's synthetic or just to kind of get you used to those terms. But I'm more interested in knowing, like, what do we use it for? You know, are there any nasty side effects that would keep us from using this? So I know I'm like throwing in a lot of details, but these are kind of what I would want you to focus on, and you could incorporate that into a concept map of your own as well. Okay, don't be afraid to stop if you have questions or comments. Okay. All right, so our next big class of antibiotics are the protein synthesis inhibitors. All right, so in general, what we can say about these, not just the aminoglycosides, but our macrolides, our tetracyclines, most of them are broad spectrum. So remember, that has advantages, disadvantages. It's an advantage if you don't really know what the organism is that you're trying to kill, because it's likely to kill it. Disadvantage is that it kind of knocks down our normal microbiota, which can kind of open the door to other undesirable things like a C. diff infection or a yeast infection. So it's really best if you know what you're dealing with to get a drug that will target just that one thing. And we talked about, you know, the kinds of organisms that secrete antibiotics. Streptomyces, that was the bacteria, the soil bacterium that kind of acted like a fungus that made reproductive spores and grew in filaments. A lot of these drugs are the natural product of Streptomyces. All right, so aminoglycosides. They are going to bind to and change the shape of that small subunit of the ribosome. All right, so that's their mechanism of action. Like I said, they're broad spectrum. They don't absorb very well, and they are degraded by stomach acid, so they have to be injected. Okay, and they're generally regarded to have high toxicity. So I found this little cute little maybe mnemonic here for aminoglycoside toxicity. You've got the kidney punching an ear, and on the boxing glove are written the names of some aminoglycosides like gentamicin, tobramycin. I can't even read the other ones because they're so small. Okay, so major toxic effects are ototoxicity, that's toxicity to the ears, and nephrotoxicity, toxicity to the kidneys. So we have to dose these out pretty carefully. So examples of drugs that are aminoglycosides, they typically end in that suffix mycin. All right, so kanamycin, gentamicin, streptomycin, neomycin. Just FYI, they spelled gentamicin different because it's produced from a different organism that's not produced from Streptomyces. So they thought they'd be cute and spell that one a little bit different. We're going to talk a little bit more about streptomycin next, but neomycin, I'll mention, this is the second antibiotic in that triple antibiotic cream. Okay, so you've got the cell wall inhibitor, bacitracin, neomycin, a protein synthesis inhibitor, and a third that's a different mechanism that we'll get to shortly. All right, so I thought we'd talk a little bit more about streptomycin. Again, this is an example of an aminoglycoside. And if you look at the structure here of streptomycin, again, you know, you don't have to be able to spit back out these structures, but you should be able to look at that and recognize that it's an aminoglycoside. An aminoglycoside, it's an amino modified glycosidic sugar. Okay, so maybe if you haven't had biochem yet, you wouldn't have known this, but these are all sugars. This is another way to draw sugars out, that kind of flattened ring structure. So we've got sugars here and amino modified. So here's an amino group, amino group, amino group, amino group. So it makes sense, the name aminoglycoside, amino modified glycosidic sugar. And streptomycin was actually the second antibiotic after penicillin that was developed. Okay, so we came out with this, I think, in 1946. Penicillin was mostly active against gram-positive infections. Streptomycin was useful against tuberculosis. So this was a huge, you know, miracle drug. We had struggled with TB for centuries, so it was kind of a big deal. And your book here has a little figure just exemplifying the mechanism of action here. So streptomycin here, it binds to that small subunit of the ribosome, changes the shape. So basically the messenger RNA is misread. And we get amino acids incorporated into the protein that don't belong there. So probably not going to have a functional protein. Broad spectrum, toxicity to ears. So I mentioned that ototoxicity and nephrotoxicity in the last slide. To me, I think, well, how can a drug be toxic to your ears? It actually acts on cranial nerve number 8. All right, so those of you that are in anatomy, maybe that means something a little more to you. I was just talking about cranial nerves with my daughter because it's this week in her curriculum in first year of medical school where they've had to use the bone saw and cut open the skull of their cadaver. She said it was just awful. Like she's got a pretty thick, you know, strong stomach, but she couldn't do it. They had to, you know, cut it in 2 ways and take the brain out of the cadaver. But she said, oh, you could see the cranial nerves, and that was kind of cool. I couldn't do the cutting part. So streptomycin acts on cranial nerve number 8, and that can cause a lot of different effects. So that can cause maybe some ringing in the ears. It can cause a little bit of tinnitus. So that's what tinnitus is. Vertigo. So you lose your bout and like you feel real nauseous. It can cause hearing loss, even deafness, and those effects can all be permanent. So we have to be very, very careful how we dose out streptomycin. And now we would never use that streptomycin unless all our other possibilities were exhausted because of the potential of hearing loss. But when it first came out, again, this was the first drug we had that was useful against TB, bubonic plague, that's caused by a gram-negative organism that penicillin didn't work against. So now we had a tool against bubonic plague. Another very serious infection is tularemia, which not common. It's carried by the fleas of rabbits. and it has the potential to be deadly. So again, it was, you know, we don't use it now unless nothing else works, just because of those potential side effects. Another one that can have some nasty side effects is tetracycline. And there's actually many different tetracyclines, or just, let's say, tetracycline in general. 4 rings, all right? So the structure shouldn't surprise you. 4 rings here, tetracycline, makes sense. It targets also the small subunit of ribosomes, but it does it in a little different way. Just skip to that graphic here in a minute. It actually blocks the docking site of the incoming charged tRNA. All right, so it's considered more of a static drug, meaning it keeps the bacteria from growing and reproducing, doesn't kill it right away. If you keep it from growing for long enough, then it gives our immune system a chance to kind of take over. So once the concentration goes down and the tetracycline falls off, then it's going to start producing, synthesizing proteins like normal. Okay, so once again, broad spectrum, semi-synthetic, really cheap to produce. So that has had, I guess it's good and it's bad. It's bad in that we've really overused it. We've used tetracycline in agriculture now for a long time. And we kind of happened on that by accident, that, hey, if you put antibiotics in animal feed, yeah, it keeps the animal from getting sick. It also makes them grow faster. So that was kind of a happy coincidence, but then it led to the massive overuse of antibiotics in agriculture. So much that, you know, we're worried that that has contributed to the problem of antibiotic resistance, you know, in treating human infections. Okay, so especially in chicken feed, lots of tetracycline in chicken feed. We're kind of moving away from that now. A lot of time, like, you know, restaurants will say, oh, our chickens haven't been fed antibiotics, and some states have rules that you can't add antibiotics to animal feed. It can be taken orally, so that's kind of a good thing. Used primarily for a few different infections in adults. You may have been on an acne medication that had tetracycline in it, prostate infections, intracellular parasites, kind of, again, just kind of a short number of infections. Never give this to children because it stains developing teeth. So that picture really jumps out at you. We want to avoid that if possible. It impacts growing bones as well. So it's okay to use in adults, but you wouldn't want to use it in children. And it also causes patients to have, it causes a phototoxicity. So what that means is you get sunburned much more easily if you're on a medication with tetracycline. So if it's in your acne medication, every time, you know, you go outside, especially in the summer, you got to have sunblock on. Interestingly, you know, drugs always have a use by date on them. And usually you want to use it by that date because after that date, the drug starts to kind of break down and not be as effective. Tetracycline, if it goes beyond the date, actually breaks down into a product that's much more toxic. All right, so always a good idea to just kind of throw your drugs away if for whatever reason they don't get used, just so you don't use them later on. And again, have it be something that's potentially more toxic. All right, so the last one of our protein synthesis inhibitors, the macrolides. Azithromycin is an example of a macrolide. The other one that used to be used a little bit more commonly is erythromycin. But these target the large subunit. Okay, so and it's more of kind of a physical block here to keep the ribosome from moving down and reading the messenger RNA. Okay, so stops protein synthesis. Again, I talked about amoxicillin being really commonly used in kids, but I think erythromycin is one of the more commonly prescribed antibiotics as well. You may have taken it as part of a Z-pack, they call it. Especially common to give to patients with penicillin allergies. All right, so kind of, your run-of-the-mill everyday infections like ear infections, strep throat, you may have taken a Z-pack for that. It's kind of nice because you only have 5 pills typically, and you take 1 a day. All right, so the activity, it just lasts a long time, so it's really kind of much more convenient that way. Also, it's one of the drugs used to treat gonorrhea. That's an infection that we'll talk more about later on in the semester. It's the natural product of, I'm not sure what organism produced that. I think it's, I don't know. I would have to guess. I'm not sure which one. Broad spectrum, but it's kind of, some gram positives and some gram negatives, not absolutely everything. There are some allergies to this as well, not as commonly as to penicillin, some GI issues. So it can kind of, kind of cramp you up a little bit, maybe a little diarrhea, kind of makes you feel like you just don't want to eat. Less so than erythromycin. When I was growing up, I always have to these little orange pills that were erythromycin. Azithromycin doesn't have quite the problems with GI issues. And you may recognize this drug. Last fall, they were talking about using azithromycin to treat COVID. And I thought, well, that's kind of unusual, right? We know antibiotics only work on bacteria, right? They pound that into our brain. So why could we use this? Why should it be used to treat a virus? And of course, we were looking for drugs that were already used for other things, because then we already knew that they were safe and didn't have to run them through any more clinical trials. It turns out azithromycin has some anti-inflammatory properties as well. So we know that's a problem with some COVID patients. They just have too much inflammation, and that contributes to the disease. So azithromycin has some anti-inflammatory properties, and we think it does upregulate the production of interferons, which is a cytokine that our innate immunity produces to help with viral infections. We'll talk more about interferons later on as well. So those two things we thought may help. I don't think that they found any difference once they studied it a little bit more carefully. I don't think it's something that's commonly given to COVID patients now, but interesting because that was just something that was tried. All right, new mechanism of action then. We have a RNA inhibitor and a DNA inhibitor that we'll talk about. Rifampin, or sometimes it's called rifampicin, it's the same thing. This is a drug that blocks RNA polymerase. Okay, so it works at the level of transcription. Okay, blocks transcription. Semi-synthetic, easily absorbed. It has a really interesting side effect. Because rifampicin, the pill itself is bright orange, patients that take it, their bodily fluids turn bright orange. So here's an example of the bag from a catheter collecting urine that's bright orange. It's kind of alarming, but it's supposed to be harmless. So there's some other side effects, like nausea is kind of a common one, as well as the orange bodily fluids, fatigue a little bit. Let's see, uses. Typically use this to treat TB and leprosy. Okay, so remember the other TB drug that we talked about, the one that targets mycolic acid, isoniazid, typically is used in combination with rifampin. Okay, that's kind of like the first 1-2 punch that you give to TB patients. Leprosy is another infection caused by mycobacterium. All right, and then the DNA inhibitor that I want to mention, and I actually left this off, this drug off the learning objective, so I'll post something to add that one back in. I decided I wanted to talk about it after all. The quinolones and the fluoroquinolones. So these both target DNA gyrase. And if you remember, DNA gyrase is that enzyme that works during replication upstream of the replication fork. It's supposed to relax those supercoils. Okay, so it cuts the strands, unwinds them so that that replication fork can keep keep going. Going this way, this way, this way. Okay, and that enzyme, remember, we're in the back of our minds, why is this selectively toxic? Of course, bacterial ribosomes, different structure than ours. That RNA polymerase that bacteria have is a little bit, it's different enough than ours. We have a different RNA polymerase to make every kind of RNA. Bacteria just have one, so it's different structure. Same thing, we have enzymes that work to relax supercoils, but they're different enough that we can have an antibiotic that targets that. Okay, if this enzyme is not working properly, then the DNA can potentially break due to mechanical stress. All right, so again, this is the DNA of the bacteria. All right, and sort of the first quinolones were kind of the first generation, the earlier drugs that targeted DNA gyrase. An example of that is nalidixic acid. That will just ring a bell, hopefully, if you're in lab, because colistin and nalidixic acid blood agar plate, that's the media that we use that only gram positives grow on. Okay, so works on gram negatives only. Okay, soon after that, we started kind of monkeying with the quinolones a little bit, and we found that if we add a fluorine group here, this is kind of then the second generations or the later ones, we call them fluoroquinolones. They had better activity against DNA gyrase, and they were a little bit broader spectrum. So just by adding that fluorine atom there. And the fluoroquinolones end in this oxycin, right? So there's a lot of different ones. They almost all end in that suffix oxycin. Ciprofloxacin is maybe one of the more recognizable ones that has the trade name Keflex that you may have heard of. So again, the spectrum of it does depend on the generation. Quinolones, basic, as far as I know, aren't in use anymore. It's mostly fluoroquinolones, at least when you're treating humans. It has an advantage of being readily absorbed and stable in acid. Uses, so we're not going to use these for run-of-the-mill infections. We save these for more serious infections, urinary tract infections, sexually transmitted diseases, respiratory infections. Keflex is the antibiotic that's typically given if somebody might have been exposed to anthrax. So you give it to them so they won't develop anthrax. Big problem with fluoroquinolones are the side effects. Some of them are severe. So the FDA came out about 5 years ago with its highest warning that fluoroquinolones actually aren't supposed to be used unless other things have failed. All right, some of those nasty side effects, kind of unusual, tendon disruption in older adults. Okay, so Achilles tendon can rupture. You might think, oh, that's kind of a weird side effect of an antibiotic. We're not really sure why that happens. It might be because it impairs collagen production. It might just limit the blood supply to joints. We're not really sure, but it's kind of a rare side effect that has happened. Some neurological things like seizures in infants, nerve damage in children, headache, dizziness, even aortic rupture. So again, some potentially really serious side effects. And again, much less use than previous. I was reading about these, and one epidemiologist said, overuse of fluoroquinolines is like trying to kill a fly with an automatic weapon. All right, so it's kind of way more, than what is typically needed in terms of, whatever. Again, the serious side effects there. And I came across a mnemonic that might help remember the mechanism there. So I thought I'd pass that along. The queen stops the gyrating dancers. So queen for quinolone stops the gyrating dancers DNA gyrase. Okay. Pass that along. All right, so cell membrane inhibitors. We don't really have many of these because bacterial cell membranes are so similar to ours that it's hard to get that selective toxicity. So there's a group called the polymyxins, and actually colicin is an example of a polymyxin. Again, if you're in lab, the colicin and the nalidixic acid target gram negatives only. So that's just an example there that you might have heard of. They work by inserting in the membrane. So you can kind of imagine with this structure, we've got this long tail here that inserts down in the membrane and disrupts the membrane, right? So anytime the membrane's disrupted, things are passing in and out of the cell that shouldn't be, energy can't be generated as easily. They're most active against gram negatives. So again, they have the outer membrane, so that makes sense. Lots of toxicity issues. So they are only used topically on the skin, okay, because of toxicity issues with the kidneys. Okay, but again, here's our third drug in the triple antibiotic ointment is polymyxin B. So the cell wall inhibitor, bacitracin, neomycin, aminoglycoside, and now we've got cell membrane inhibitor. So kind of a 1, 2, 3 punch. Okay, so good for skin infections, really too toxic to be used for much else. All right, lastly, our last drug and our last mechanism of action. We have a few examples of drugs that target a metabolic pathway that bacteria have and that we don't. Okay, and this is a class of drugs called the sulfa drugs. And actually, the drug that we're going to talk about here, SXT, that stands for sulfamethoxazole. So we get the S and the X here, and trimethoprim. So we often use these two drugs. We use them together because they impact the same metabolic pathway, right? So again, you don't need to know the specifics here, but in generally, this pathway, we start out with this para-aminobenzoic acid, or PABA. Sulfa drugs inhibit kind of the first enzyme in that pathway. There's an intermediate here, a second intermediate, and then a third enzyme in that pathway is inhibited by trimethoprim. Okay, so these drugs are inhibiting the same pathway. This is the pathway for bacteria to manufacture folic acid. So tetrahydrofolic acid is essentially the same thing as folic acid. Okay, and bacteria need folic acid in order to synthesize DNA or nucleic acids. Let's say nucleic acids. Okay, so you keep them from synthesizing their DNA and they're not going to be able to reproduce. Right? So when we use the sulfamethoxazole, so all the sulfa drugs have these sulfonamide functional groups here. We use that together with the trimethoprim, then it's really, again, kind of a 1-2 punch. The trade name for this is Bactrim. So you may have heard of Bactrim. Okay, so the graphic that your book shows, it just shows it a little bit differently here. Here's that same pathway. Again, you don't need to know what the enzymes are in between there, but it's a pathway to end up with folic acid here, which again is necessary to make DNA and RNA. So the way these drugs work, here is what normally should happen. Okay, here we've got the enzyme in the pathway, and this is the substrate that's supposed to be there, para-aminobenzoic acid. And what you end up with eventually is folic acid. So that's a bit of a simplification, right? There's like more steps to that process, but that's the way the latest edition of the book shows this. All right, so what you notice here at the top, here's the structure of para-aminobenzoic acid, and here's some examples of different sulfa drugs. But what you hopefully notice here is that the part of those sulfa drugs all looks exactly like part of the substrate that's supposed to fit in there. All right, so the sulfa drugs, so here's a sulfa drug over here that fits into the active site of that enzyme where the para-aminobenzoic acid is supposed to fit, but now can't. All right, so then you don't get folic acid. All right, so essentially this is an example of competitive inhibition. Okay, so the antibiotic is binding to the active site so that the substrate can't bind and you don't get the product. Okay. If the concentration of the sulfa drug diminishes, then more substrate would be able to bind there and folic acid could be produced once again. Okay, why is this selectively toxic? Don't we need folic acid in our cells too? Yes, we do, but we get ours from the food we eat. We don't make our own. All right, so again, it's a metabolic pathway that bacteria have that we don't. Okay. All right, so hopefully, go back in and at some point fill in those other antibiotics that we talked about in the concept map there. And again, hopefully that will help you when you're trying to pound things into your brain there. All right, so one other topic that I want to talk about, of course, is antibiotic resistance. We have a number of people over in the chemistry department that do research with antibiotic resistance. So if that's a topic that interests you, I can point you in their direction. This is a huge problem. This is one of those, problems that we certainly worry about, that antibiotics will eventually no longer be effective. And of course, it's taken kind of a backseat with the whole pandemic, but it's not going away. We are not coming up with new antibiotics at a rate that's going to keep up with the bacteria that are evolving ways to withstand them. So we'll think a little bit about kind of in general how bacteria become resistant. We'll think a little bit about what we can do about it. And then I want to look at kind of the particular mechanisms that how do bacteria actually enable themselves to live in the presence of antibiotics. So there's just 3 or 4 different mechanisms specifically that we'll talk about there. So in general, how do we think antibiotic resistance has come about? Well, in any and this is a graphic that I pulled off the internet. I didn't cite that very well. I think I had trouble finding it again when I went back to cite it. But this is not from your book. The theory goes that in any population of bacteria, you have kind of wild type cells that wouldn't have any reason really to be resistant to an antibiotic. So mostly they're non-resistant. But because bacteria multiply by the billions, and they're going to mutate now and then, you might occasionally get a mutation that makes the cell able to survive in the presence of antibiotic. Okay, so the yellow ones here are the cells that have somehow either acquired the ability or have, in this case, some mutation in their DNA that allows them to live in the presence of antibiotics. Normally, those cells would not be as fit as the other cells. So normally they would probably not grow quite as quickly, and they would just, kind of die out. But if there is some selective pressure, meaning if the cells are in the presence of antibiotics, then any cell that does not have that mutation is going to die. And only the cells that have mutated it to be able to live are going to survive. And then they're the ones that are going to reproduce. So the selective pressure is going to exist mostly in hospitals. Patients in hospitals, they are almost all on some kind of antibiotics and probably more than one. So hospitals is where we often see these antibiotic resistant infections showing up. So some examples, we've already talked about MRSA. We mentioned vancomycin resistant enterococci. Huge problem with strains of Mycobacterium tuberculosis causing drug-resistant TB, gonorrhea. We keep having to change how we treat that because strains of gonorrhea keep becoming resistant. So it's just a huge, widespread problem. Like I said, that's not going to go away anytime soon. So what do we think have contributed to the resistance spreading? Like I mentioned, that just natural selection, the selective pressure of using a lot of antibiotics. The more often we use them, the more likely that their mutations are going to help them survive, and especially in healthcare settings. We know that at least more so in the past, but even now antibiotics are misused. Patients demand some kind of drug to make them feel better, even if it's potentially, probably viral, and the antibiotics won't do any good. Sometimes doctors just will prescribe something to get patients off their back, or, parents that say, give my cranky kid something to make them feel better. And they, it happens still, probably less today, but it still happens. They've been vastly probably overused in agriculture as a preventative, just to increase their profit margin. Profit margins are slim. I can't blame them, but it's something that we're trying to get away from now. And worldwide, in other countries, you can buy antibiotics just off the shelf. You don't need a prescription. So people take the wrong dosage, or especially they don't take as many as they should to totally, get rid of the infection. So that's a huge problem. It's not surprising that a lot of our antibiotic resistant infections kind of have started out in poorer countries of the world where, again, their antibiotic use isn't regulated very well. So what can we do to try to slow down resistant? Something that you can do yourself and encourage everybody that when you are prescribed antibiotics, take the full prescription. Sometimes, the antibiotic, can make you a little nauseous or whatever, and it's easy enough to say, well, I feel better, I'm just not going to take the last couple of days. You want to make sure that you kill all of those bacteria. So there's always a few more, most of them are killed off the first few days, but there's always going to be a few more that you want to take your full dose to make sure that you're rid of them. My father is the worst at this. He is prone to gum infections. So he gets an antibiotic from his dentist and he takes most of it, but then he saves a couple days worth for the next time he gets an infection. like, yeah, we think that might contribute to organisms developing resistance. So you really don't want to do that. And this is kind of a hard one. As much as you can, don't take antibiotics. And you should let your doctor decide whether you need to take them or not. So I see on Facebook, I have a friend that, oh, she thinks her kid has strep throat. I'm going to have them gargle with cider vinegar or something for a few days. No, strep throat has potential complications that you don't want to deal with. Get on the antibiotic for strep throat and get rid of it. But for some infections, like ear infections, now they're saying that like most of the time those clear up on their own, and without antibiotics. So it's a tough one because, you know, kids are cranky because of it or in pain. You want to get rid of it as soon as you can. So again, that's kind of a tough one. We have tried in treating a lot of different infections to use a couple of different antibiotics at once. So we know that sometimes there's a synergistic effect. When you use two antibiotics, they have an even greater killing power than, you know, either one separately. So you can see this on this, looks like a Kirby-Bauer test here. The zone of inhibition is greater where both of those antibiotics have, you know, diffused out to meet one another. And this can also, if you hit a bacteria with different antibiotics that work in different ways, that can kind of slow down the possible resistance acquisition as well. So just some examples. We talked about that augmentin, how amoxicillin is almost always marketed now with clavulanic acid. SXT is that trimethoprim and sulfamethoxazole. When we treat TB, we always give 2, usually 4 drugs at once that all work in a little different way to try to keep that resistance from showing up. We are constantly kind of messing with drugs that we know are safe to try to kind of stay ahead, a step ahead of any resistance forming. So, you know, the semi-synthetics, we're kind of monkeying with some R group. And how they do that, that's all organic chemistry. I couldn't really tell you how we actually produce those drugs. A few years ago, somebody came to Grand Valley giving a seminar. She was talking about tuberculosis, and her whole research was targeted towards finding novel proteins, particular in Mycobacterium tuberculosis, so that we could design a drug to treat that protein that only the tuberculosis had. So you design a drug specifically, you know, for one organism, very narrow spectrum, and that again should slow down resistance. Okay, so how do bacteria acquire resistance? Sometimes by mutation, but most often by some kind of horizontal gene transfer. So little review, remember the 3 kinds of horizontal gene transfer. This figure is a little confusing until you really look at it. But remember, transformation, that's when a living cell will take up some DNA from a cell that's dead. Cell walls breaking up and the DNA can leave that dead cell and enter a live one. Transduction involves some kind of virus kind of accidentally taking part of the genome of one cell and transferring it to another. And then conjugation is when a cell would pass on a plasmid via a conjugation pelus. Okay, so we think that especially conjugation is probably an important means that cells share their ability to be resistant to antibiotics. All right, so next we're going to look at the 3 different, well, 3 or 4, depending how you count it, different mechanisms of resistance. So what specifically a cell can do to keep it from surviving in the presence of antibiotics. And I think we'll finish up with that next time. We'll get into chapter 14, looking at process of infection, and next week start immunology. So, all right, have a good day. Stay warm.